Good morning, everyone. I am welcoming you all to this 19th day of CSIR Life Science Question and Answer discussion session. Today, we are going to discuss about the methods in biology. Yesterday, we had some part, okay, that is the part C important portion, uh, like 13th unit question and answers. Okay, once again, I will tell the in the part C of CSIR examination, they are covering mostly 20 questions, nearly 20 questions from the unit 4, 5, and 13. You need to answer 25 question in part C. Among that, there is nearly 20 questions are there in that from 4, 5, and 13th unit. Just remember it. So just understand the concepts of the unit 4, 5, and 13. Previous days we discussed the unit 4 and 5. Today we are going to discuss about the unit 13. Like here you can see the methods in biology. Some all the concepts are comes under the methods in biology, like biochemistry, protein engineering, molecular biology, bioprocess, food processing. All those experiments are comes under this category. So we will enter into the session. A protein is composed of leucine, isoleucine, alanine, glycine, proline, one lysine, one arginine, two cysteine connected by the disulfide bonds. Okay, these are all the compounds of the protein. That structural, just the protein structure having the leucine, isoleucine, alanine, glycine, proline, one lysine, one arginine, two cysteine connected by the disulfide bond. Conformational analysis indicated that the protein has elements of helix and beta structure. The protein most likely not an enzyme. How they tell this is not an enzyme? Okay, here the notable thing here is the conformation analysis indicated that protein elements of helix and beta structures. Okay, they are having the helixes and beta structures. Okay, why here what we want to understand, the enzymes are having the helical structures. Full structures are made up of the helical structures. So here they mention it's a elements of helix and beta structure. Beta sheets are there. So as per their statement, this enzymes, okay, um, that this protein, the protein is not a enzyme, so the output is it's not a enzyme. Here the notable thing, one more notable thing, enzymes in the they are mentioned, they are made up of disulfide bridges. Normally the if the enzymes having disulfide bridges means they are not be flexible, so they need a they are make a covalent interaction with substrate molecule and other linked molecules. So first statement is just the information. The key statement is the conformational analysis indicates the uh, indicates it's having the protein, it having the beta sheets and alpha helices. Okay, next concept is very important concept in the NMR. Hydrogen atoms in a delta methylene group of lysine will give the following splitting pattern in the NMR spectra of lysine. Each and every molecule is given separate band, separate spectra. Okay, splitting pattern is different for each and every atoms. So here they asked hydrogen atoms in the delta methylene group of lysine will give the following splitting pattern. What pattern they are provided? They are providing the triplet and triplet of triplet. Okay, triplet of triplet pattern. In this case pattern, they are specifically recognized as the this lysine, sorry, in this meaning of triplet means they having the that that illustration, that pictorial illustration of NMR spectroscopy, the a signal that has been splitted into the double, and the each lines of double splits into triple. Okay, one peak is divided into two splits, that is the double split. That double splits, okay, the lines of this double splits are into triplet. Okay, that is called as the triplet of triplet. That peak also divided into triplet, so only it's called as the triplet of triplet. This lysine structure, that hydrogen atoms which present in the delta methylene group of lysine, we are having the triplet of triplet. Okay, in this case, they each in a, another case. Okay, this quintet. Okay, this quintet is another split concept. In this concept also, they are having the various ranges. Okay, splittings, that quintet are having the that are also known as the pentet. Okay, pens kind of quintet are known as having, they having the 5 lines, 5 split ends. NMR is a fully all about the splits. First one, lysine, is known as the triplet of triplet. In that case, one peak is divided into two peaks. That two peaks are divided into triple peaks, that is triplets. They are having a split. They are having a line. That line is called as the triplet. In this quintet, they are having the 5 lines, 5 lines that are closely together. This NMR splitting is vary for that are vary for various molecules, but this character is helpful for identifying the atoms. Okay, then what is for example, the DMSO, dimethyl sulfoxide, that's dimethyl sulfoxide, that given this kind of peaks. This is one example. Here the lysine gives the normal triplet lysine, that methylene group of lysine give the that triplet of triplet splitting. DMSO giving the quintet splitting. Double of double of triplet, triple of doublet, that are given another case that are different from the this normal conditions. Then okay, normal triplet. Okay, some normal triplet form means one peak is divided into two peaks, so that splittings is known as the, it's having 3 parts, various regions. In this case, they are that deuterium is known as the called as the triplet in the NMR. The deuterium compounds having the NMR speak with triplet splittings. Okay, next concept is the combination of molecular markers with their classification based on their dominant and codominant types are shown below. Okay, this is all about the combinations of molecular markers. Molecular markers means it's identifying based. The classification based on their dominant and are here they are mention the SSR and RFLP. They are codominant. SSR and RAPD, it's a codominant, and RAPD and RFLP, that are dominant. AFLP and RAPD is a dominant. Which one is the correct combination? Here, before enter into the answer, we are all want to know the what are all the that RFLP, that concept. Here, that is first thing, that the SSR is the thing which is used in the simple sequence repeats. Okay, simple sequence repeats, short term repeats, that known as the short term repeats, which is the marker. Okay, which is the marker. In various genetical studies they are using this marker, that known as the short term repeats and simple sequence repeats. Another one is the RFLP. This RFLP is the restriction fragment length polymorphism. In this restriction fragment length polymorphism, they are using the restriction enzymes in the and do the experiment. They are also another markers for the genetic mapping. Okay, next is RAPD. This RAPD is a well-known concept, that is the random amplified polymeric DNA. First one is the restriction fragment length polymorphism, is RFLP. Another concept is the random amplified polymorphic DNA. Okay, likewise, this another term is FLP, this amplified fragment length polymorphism, that is amplified fragment length for polymorphism. In this RFLP, what they doing means that bacterial that experiments, some experiments, they are finding the variations in the DNA sequence with restrictions enzymes, so they can create a map. This is one marker. Here they are want to first keyword is they are want to their classification based on the dominant and codominant. Okay, here the correct concept is D answer is a P and S. The short term repeats and the RFLP, that is a restriction fragment length polymorphism, is a why they are mentioned as a codominant. If while you are using the restriction enzyme, that whole length of fragment is divided into small pieces, that small pieces are the short that having the similar length of short uh, simple nucleotides. Okay, short nucleotides, short term repeats, so they are codominant. Another case is RAPD and RFLP. This RAPD is concept is known as the random amplified polymorphic DNA. That amplified DNAs, polymorphic DNAs, are randomly amplified and stored in a library. In RFLP, restriction fragment length polymorphism, here that restricted, okay, particular restricted DNA that having a that having a similarity between whole all the organisms length of genes are stored, so these two are the dominant. Restriction fragment length polymorphism and random amplified polymorphic DNA. Only one tryptophan residue, that is W, which is buried means it's inside of the protein. The protein is cacotylated buffer. Cacotylate buffer is excited at 250 nanometer, and its emission spectrum measured in the range of 300 to 450 nanometer. The sum the same measurement was repeated on the proteins in a buffer containing 6 molar guanidine hydrochloride. It was observed that there is an increase in the density of emission spectrum of guanidine hydrochloride treated the cytochrome C, that increase the intensity. The most probable reason is uh, W is near the heme in the native protein. Okay, heme means it's nearly in the active center. Here the concept is the protein, after that this buffer added to the protein, that will get activated and enter into the center part. Okay, here a next concept on the this molecular biology technique, it's known as the that restriction enzymes used and restriction enzyme cutting regions. The diagram below represents a 2 kb insert successfully introduced between two BAM1 site of 3.8 kb vector in the desired orientation. The hint 3 site on the insert and EcoR1 site on the vector is also indicated. If the insert was introduced in the opposite orientation, which of the following statement is incorrect? So they are provided the statement, they are provided the image, you want to find out the incorrect. First step, okay, they are mentioned, the diagram below represent 2 kp insert successfully inserted between the two BAM1 site of 3.8 kb vector. This is the 2.8 kb vector. The whole round is known as 3.8 kb vector. The BAM1 site is two sites are here. So the inserted molecule is below this 2 kb. This 2 kb is inserted between the BAM1 site. Hint 3 site on insert. The main important word here is hint 3 site on the insert and EcoR1 site on vector also indicated. Here the hint 3 site here and another site is EcoR1. They are provided the full information. So then what we want to understand, if the insert was introduced in the opposite orientation is inserted in the opposite, okay, this is the BAM1 side. If the insert is it's loaded in this area means, okay, main thing, if that is inserted in this area, here first one is direct orientation, opposite orientation means this is also the site between the BAM1. If it is bind in this side, which is the incorrect statement. Digestion with EcoR1 will linearize the 5.8 kb plasmid. Why they are mentioning the 5.8 kb? Digestion with EcoR1 means you will get cut at this particular area. After getting the cutting in this area, that is become linearized. So this having the 3.8 kb. Additionally, they are added 2 kb insert that is also present in the particular region. So the total is kb is 5.8 kb plasmid. That is one possibility. Another possibility, digestion with BAM1 with yield 1 3.8 and 1 2 kb fragments. If there is a binding, okay, that compound 2 kb is bound at this particular area, this digestion with BAM1 means that will give a 3.8 whole plasmid and another fragment is 2 kb fragment. Then another statement, right statement is a D. Double digestion with EcoR1 and hint 3 will produce the 2.1 kb and 3.7 kb fragment. This is very interesting concept. Why it gives the this answer? They are added some 2 kb insert here. Okay, after the 2 kb inserting, what they did means here the loss of their digestion with the hint 3. So for hint 3 here, that is to one region is remaining with the 1.2. Here this region is 1.2 kb and another thing, that 1.2 plus this 2.9 kb. Here its remaining part is the hint 3 cut only 1.2 kb. this 1.2 kb plus this 2.9. So totally it will get 3.7. And another case, the eco-R1 cutting, if they are digested with eco-R1, they will get 0.9 plus 0.8, that is known as the 2.1 kb. These are all the possibilities. So incorrect statement. What is incorrect statement is digestion with eco-R1 and Hindipti will produce 1.7 and 4.1 kb fragments. It's a wrong statement. Why it is wrong statement means we want to analysis, we want to create the digestion of eco-R1 in the particular region called this particular region. They are adding the whole okay, that, sorry, they are adding this particular portion alone. For the digestion we want to insert this full portion 2.9 and the 1.2, then only we will get 2.1. Don't need to add this 0.9 and 0.8. In this case we want to, they are the wrong statement, they are mentioned 0.9 and 0.8 is a possible addition, that is wrong. So whenever you are doing digestion, that addition of this 1.2 and the this whole region and addition of this 0.9 plus 1.2 is a whole region, that is the right answer. And next concept in the methods in biology, given below are some of the methods used to access evolutionary phylogenetic relationship among the plant taxa. It's very important thing. Some of the methods used to access the evolutionary phylogenetic relationship. Its main concept, the phylogenetic concept in the among the plant taxa. What they are used means 16S rRNA. This concept is well known for many microbiology peoples. Uh, and this 16S rRNA sequencing, what they are doing means they are in this sequencing method. They are collecting the DNA molecule, and that having the specific gene is called as the 16S rRNA. That gene has been isolated from particular sequence, and then they are doing that that sequencing process, that based on the particular gene expression they will get um, the characters, the nature of the particular sickness or particular species. In this 16S rRNA, the very important thing that which compound will having which cell which having the 30S ribosomal subunit. Okay, the main thing, the cell which has the that 30S ribosomal subunit, that thing only helpful for the this 16S rRNA sequencing. This to identifying this plant taxa genome, they are doing this 16S rRNA sequencing. And next thing is a mitochondrial satellite. It's in this mitochondrial satellite concept is a new method. In this latest researchers, okay, mitochondrial microsatellite in method is the latest method which reveals the maximum number of small length the maximum number of the species. Okay, in this microsatellite mitochondria, they are using the nearly it's 20 to 60 base per length sequence of DNA, and finding their specific regions, specific characterization regions. In this satellite, that they are the mitochondrial microsatellites are very very specific for the species. The once you are finding the species, you can easily identify the taxa. These two things are very important. This both things, okay, this 16s rRNA sequencing and this mitochondrial microsatellite DNA, that microsatellite concept, both are relevant to the mitochondrial DNA and cytochrome B, that 16s 30s subunit. There's a core-linked one. Okay, next. So these two things alone helpful for the evolutionary phylogenetic relationship. Okay, main thing that to access the evolutionary phylogenetic relationship, we want to do the genetic engineering molecular level study. That study is the 16s rRNA sequencing, that is the 30s ribosomal based one, mitochondrial microsatellite, that is the our small sequence, small specific sequence in the particular species. Okay, very interesting thing about next concept, it's the using of FRAB. Okay, FRAB, the fluorescence recovery after the photo. This is the flow that a FRAB method. FRAB is another. Many of them may know this antioxidant assay. That all things are having the FRAB method. In this, in the FRAB method, they are having the they are using the photo bleaching method. Okay, fluorescence recovery after the photo bleaching. This method determine the originality. They are helpful to find out the original image. The small region of surface and the particular volume containing a mobile fluorescent molecule can be recognized by the particular instrument or sensors. In this method, it's used to analyze the mobility of individual lipids within the cell membrane. Okay, lipid membranes which present in the cell membrane. So we will do the concept. This technique, diffusion coefficient of three integral membrane proteins M1, M2, M3 in the kidney cells are calculated as the 0.05 μm per s and 0.005 μm per s respectively. So this membrane proteins, three proteins are there. They're having a different uh, uh, different cell counts. Considering fluid mosaic nature of biological membrane and relationship of structural organization, integral membrane protein with the diffusion coil having the highest number of integral protein. Here the important thing they are mentioned, here the statement they are provided the information, that information is the coefficient of three integral membrane proteins, that is the coefficients are 1 μm per second, 0.05 μm per second, 0.005 μm per second. So it's considering fluid mosaic nature of biological membrane and relationship of structural organization of integral membrane protein with the diffusion coefficient. So diffusion coefficient, here they mentioned the keyword diffusion coefficient, which protein will highest number of integral protein. Okay, here the higher number of integral protein means it allows the minimum quantity. So there are tight junctions. So tight junctions means that highest number of integral membrane domains are there in the M3. So that integral membrane protein, which the M3 membrane protein having the highest number of integral protein, so only they are allowing the small amount of the molecule, small they are allowing the one, they are allowed to move only small quantity of the molecules, so only it's providing the 0.005 μm per second. That is very slow because that is the junction is too tight. So M3 only having. Okay, the resting cell, in resting cell, protein X and Y are localized in the cytosol. So two keyword here, they are protein X and Y are localized in cytosol. Upon stimulation with lipopolysaccharide, okay, it's that stimulation with the lipopolysaccharide, Both of them are phosphorylated and translocated to the nucleus. You have used antibody against phosphorylated forms of protein X and Y, which are conjugated to either red or green or blue dye. Okay, they are after the phosphorylation form the proteins X and Y, which are conjugated either red or green or blue. Keeping optical aberration of light in a mind. This is another the question statement here is keeping optical aberration of light in a mind. Which of the following will be the best for visualization X and Y in the nucleus by fluorescence microscopy? So this is the question statement is the they are provided the information. Okay, first it's present in cytosol. After the lipopolysaccharide assimilation, both of them phosphorylated and translocated. Yes, so X and Y are translocated to the nucleus. You have used antibody against phosphorylated X and Y. They are using the antibody to recognize the X and Y protein. That is either dyed with the dye is color may be the red or green or blue. This may vary for the proteins. Here they are asking, keeping the optical aberration of light in a mind, which of the following statement is the best visualization X and Y in the nucleus? By in the main thing they are in the nucleus. So which method is the best one? Anti-red X and anti-red blue Y. Okay, this anti-red for X, anti-blue for Y. This is the fluorescence microscopical representation. This is the possibility of the entry of the light. Protocol based one, it's the easiest one. The following the below are experimental protocol to find out exact location of repetitive DNA sequence and mitotic chromosome by fish, the fluorescence in situ hybridization method. Which of the protocol is given below is a correct one? So we want to study the fish protocol. The fish protocol is a fluorescence in tissue hybridization protocol. Once you have studied well about this fish protocol, you can answer this easily. The first thing is C is the correct answer. First step in the fish is the mitotic chromosomes were fixed in a glass slide. Mitotic chromosomes were fixed in a glass slide. Fixed on the glass slide. Then denaturated. After the slide fixing, denaturation. Incubate with biotinylated satellite DNA. Incubate with biotinylated satellite DNA. This is for the attachment, that complex formation. Incubate with the fluorescently labeled avidin, because first step we are added the biotinylated satellite DNA. That DNA is incorporated with the mitotic chromosome. So this complex, the mitotic and biotinylated satellite DNA complex added to the avidin. The avidin is specific for biotin. If there is a binding happen between the mitotic chromosome and the biotinylated DNA satellite, that avidin will bind at the particular portion. Otherwise here in this, after this step, this biotinylated addition, after the addition they doing the washing also. In this washing case means after the washing the non-linked are removed from the sample. So in this step the addition of adding is only for bound biotinylated satellite DNA with mitotic chromosome. This localization observed under the fluorescence microscopy. Localization observed means that is the complex. So this complex carries the mitotic chromosome, biotinylated DNA, satellite DNA, and avidin complex. Okay, next is this easiest method, easiest concept. Yesterday also we discussed this concept. A protein D is encoded by a gene which having the 5 kb long, has 3 restriction enzyme site. One protein 5 kb size which having the 3 restriction sites. The first one is 0.5 kb from a transcription start site. The first one is 0.5 kb start site. The second one is the polymeric one. The second one is the polymeric one. In order to find out whether the fetal cells containing the normal and mutated gene, total genomic DNA from fetal cells were isolated, completely digested with a hint 3. So here keyword is completely digested with hint 3. Separated as agarose gel. In this agarose gel transferred to membrane detected by the probe against region between site. So the probe against second and third restriction site, not in the first site. So it can't be recognized the first site. Here the key concept is, here they are transferred to the membrane and detected by probe against region between second and third restriction site. So we can't site 1 and the region between 1 and 2. Which of the following band pattern will be obtained in the fecal heterozygous? The important thing here they mention, the first one is a 0.5 kb. Okay, just imagine, draw the line and draw the line 5 kb length. 0.5 kb is the first side. After that after the 0.5, 2.5, third one is in the 0.5 internal to the stop codon. So this is the two things. This two second site is a polymorphic one. After the digestion, they will get this B band. Why they are getting the 3 band means, the explanation is, first step of the their experiment, they having 0.5. Okay, 0.5 is the first restriction site. In that step itself, they are having the first digestion, but not able to recognized by the particular detector. Okay, the probe is not detecting particular first region 0.5, it's not able to recognize. Okay, second the second is so the second region alone can be recognized. So second site is between, also one between 2.5 kb, it's a first site, and the from the first site. So if they are cutting between the 2.5 stage, so the both sequences are having the 2.5, 2.5, that remaining regions are having in the 2.5, just eliminate the 0.5, because that 0.5 is not able to recognize by the probe. So 2 kb is there. After this 2 kb, they mentioned that having the polymorphic nature, that having a similar nature. In this restriction to the 2.5 kb, that remaining for 0.5 is the third one. So in this also we want to remove from the that first direction. First imagine first 5 kb sequence, that second, that hint 3 is cut down at 2.5 region, that is the away from the 0.5. So that is totally 3 kb. That 3 kb we cut down the having 2 kb, another side we are having 3 kb. So this is the concept here. One side that having a 2 kb, another side is having a 3 kb. Okay, here that see this is the first step, digestion. After the digestion, the probe is not able to recognize the sequence, that is known as 0.5. They are not able to recognize. So this in this region, that 2.5, in the 2 they are not able to recognize 0.5 kb. So that is 1.5 kb is present here. So another is the the another side is the 3 kb. That 3 kb they mention here, that is the second site is the polymorphic. Polymorphic means they are shown the multiple repeats, that multiple repeats are overlapped with other sequence. So this overlapping it's mentioned, and the addition of the restriction site 1 and b, 1 and third. Additionally, they are having 1 kb, so 3 kb plus 1 kb it's become 4 kb. Next is the unknown peptide was isolated from the leaf of medicinal plant and purified to homogeneity. The peptide did subjected to Edman degradation concept is very important, and this Edman degradation is very important concept in the this plant extractions method. In this Edman degradation, uh, you can see the plants. Okay, it's amino acids into peptides. Their peptides are divided. They are sequencing the method of sequencing amino acid in the peptides. So how they are developing in this process, they are first the purifying of protein by sequential removing one residue at a time from the amino acid end of peptide. This is very important thing. The main thing in this concept, Edmund degradation, this N terminal is the removal of amino acids happen in the N terminals. Okay, first we want to understand what is the statement, what is the concept, what is the method. So Edman degradation, we want to get the basic understanding of the Edman, in which uh, the method can label and cleave the peptides from N terminal without disturbing the peptide bonds. The very important thing, it can label or cleave the peptides from N terminal without disturbing the peptide bonds between one the other amino acid residues. It won't disturb the peptide bonds. This so here they are mentioned. However, the tryptic digest of peptide produced ionic sequences mass of was found to be 18 Da less than that of obtained from the trypsin treated sample. The possible interpretation of the above experimental results less in interaction between the peptide bonds. And another which the statement is yes statement, the peptides are cyclized by peptide bond formation. Peptides was cyclized by the peptide bonds formation between alpha amine group and carboxyl group. Okay, the gene novel transcription factor. The following technique may be used to identify the cis regulatory element in the 1 kb promoter sequence of the gene where the novel transcription factor binds. Where it is binds means here the keyword is novel transcription factor regulated by the novel transcription factor. This following technique is identifying cis regulatory elements. Okay, this cis regulatory elements is a keyword here. This many of them may heard about the this cis regulatory elements. This is nothing but it's a region of non-coding DNA, the region of non-coding DNA, which regulated the transcription. So only they are mentioned the it is a novel transcription factor. They are cis elements. Okay, this is regulatory sequences. Thing that they are enhanced, they are act as enhancer. They are promoting the activation physiological regulations of the sequencing. Okay, so main thing it's element, it's or no, it's acting a non-coding DNA region. It's helpful for the transcription. So these particular element in the 1 kb promoter sequence. How to find out the non-coding sequence? So this non-coding sequences are identified by DNA's one footprinting analysis. The main thing, how to identifying these particular transcription factors, that is the DNA's one footprinting analysis. Next is a basic analytical question, is logical question. Frequency distribution of tree height in a two forest. These are all the low rainforest, this high rainfall forest area, different annual rainfall. Okay, that two different plants are there, radius are there. It's a situation, low rainfall and a high rainfall. The tree height is vary. Okay, the frequency is high, the height is become high, the frequency is become low. If the height is increased, frequency is low. Which of the following statistical analysis will be choose the test whether rainfall has effect on the tree height? The here the keyword is the rainfall has any effect on the tree height. That is the B, non-parametric comparison of two groups. It's a very important thing, non-parametric comparison of two groups. Okay, this is all about the logical statistical evaluation. As I mentioned, whenever when you are going to enter into the CSIR this examination, you want to understand the concept in the statistical probabilities. The both concept is very important. I welcome you all to this tomorrow class, that is day 20 class. If you have any questions, you can post in chat box.